I passed around handouts what the object and what it looks like.
Yeah, thank you very much for your kind introduction and thank you for the opportunity of getting this talk here in Tuorlo. So I am going to talk about very simple things actually, the stability of RNA, how stable is RNA. And mainly I am going to pay attention to the effect of base sequence and secondary structure on the stability of phosphodiester bonds, or in other words, how much chain folding actually can influence on the reactivity of phosphodiester bonds. But I think before that we somehow have to understand how RNA is cleaved and why RNA is cleaved. And it has been known about 50 years already that RNA phosphodiester bonds actually undergo two reactions, so which people very often forget. So the phosphodiester bond may be cleaved so that the 5' linked nucleoside goes and the cyclic phosphate is formed, which is then cleaved to monophosphates, a mixture of monophosphates. But there is another reaction, namely isomerization of a phosphodiester bond from the natural 3', 5' bond to the 2', 5' bond. And here is a so-called pH rate profile for these processes when we have an isolated phosphodiester bond. So we have only one phosphodiester bond present. And as you can see, so this is the cleavage. The cleavage is both acid catalyzed and it is base catalyzed, and already under neutral conditions at pH 7 it is somewhat base catalyzed. But there is this isomerization reaction, which actually under acidic conditions is a bit faster than the cleavage reaction, and under neutral or mildly acidic conditions considerably faster, about 1 order of magnitude than the cleavage. So this means that chemically, if you have just RNA in water, it is cleaved, yes, but even more rapidly, it is isomerized. And it doesn't remain as the same polymer. And I think this is one of the things which argues against the possibility that RNA could store a huge amount of information if it is not somehow packed or protected with something which retards this isomerization reaction and also the cleavage reaction. So here is the same picture once again. Why it is so complicated, I don't want to go into the sorry, now okay. Why it is so complicated? It is so complicated because the phosphate bond can be either deprotonated or it can be protonated. The sugar hydroxy function can become deprotonated at very high pH, and the phosphate may become protonated at in very acidic conditions. I don't go into details with this. And there are two reactions when we are talking about the stability of RNA which plays a role. There is this base catalyzed reaction which is quite simple. So the hydroxy function is deprotonated. It performs a nucleophilic attack on the negatively charged phosphate and the nucleoside goes. But there is another possibility, a pH-independent cleavage, which under neutral conditions already is slower than this one, and it goes in a bit different manner. So there is a proton transfer now from the OH to phosphate and nucleophilic attack at the same time, and now we actually obtain an intermediate which is more or less stable. So here this intermediate is very, very unstable and nothing else can happen. Because, so now I have to explain you one thing which is quite crucial. So at carbon atom, when we have a displacement reaction, something comes and something goes. But with phosphorus, it is usually so that something first comes, and the tetracoordinated species is converted to a pentacoordinated species, to a phosphorate, and then something goes. And now the geometry of this pentacoordinated species is like this. It is trigonal pi pyramid. And these bonds here are shorter than these two bonds. And so what comes always to the so-called apical position, and which leaves always through the apical position. So that these three ligands never can be cleaved. But it is possible that the so-called pseudo-rotation takes place, so that one of these things remain equatorial, then these two become equatorial once again, and these two become apical. How to draw it? So something like this. And now here, because these oxygens are negatively charged, they can never take an apical position. This pseudo-rotation is not possible. It can only cleave. But here, when we have an OH, it can take an apical position, and this pseudo-rotation indicated here is possible, and isomerization occurs. So this is a peculiarity of phosphorus chemistry. I skip this, so we know that the transition state is very late and so on, but it is not so interesting for you. I think not even this either.
I mean, does this mean that phosphorus polymer nucleotides are, because of this phosphorus properties that you just explained, unstable or easily destabilized?
No. No, that does not make it easy, but I think in RNA the reason of the stability is the 2' hydroxy group, which happens to situate it in such a way that the reaction is possible. I will come to this in a moment. So what I showed was done with small molecule models, isolated phosphodiester bond. But even if we go to a polymeric structure, nothing is actually changed. So the individual bond usually still behaves as I described. If there is no secondary or tertiary structure, if we have a simple polyuric acid, polyU, so it behaves like that. But now, if we have an interaction between the bases, as for example in double helical nucleic acids, as in double helical DNA, also RNA can be double helical, which means that between the bases there is a stacking interaction. They partly tend to overlap each other. It means that when this crucial reaction site, here the attacking nucleophile, phosphorus, and the leaving group, which, because the leaving group can go through apical position only, must become collinear before the bond can be cleaved. So it means that to cleave this bond we have to destroy the base stacking here and end up to this kind of open structure when it cleaves. And if we have a double helical structure, so this base stacking is so strong because it is not base stacking between two different bases but two different base pairs. And in that case, so it needs a lot of energy to destroy this base stacking, and that is why double helical RNA is much more stable than single-stranded RNA. But in single-stranded RNA, this still may happen. So why then also in single-stranded RNA we may see rate retardations, that some phosphodiester bond is not cleaved as rapidly as in polyuridylic acid. So also, as I mentioned, in single strands there may be a base stacking, which forces the geometry unfavorable to the cleavage. The 5' linked nucleotide cannot adopt this kind of apical position here, needed for chain cleavage. And of course it is possible that the cleavage takes place, but the nucleoside is not here apical as it should be to the cleavage, but owing to base stacking it adopts an equatorial position. And now, since these are both negatively charged, it cannot pseudo-rotate, and no cleavage takes place. And there is a considerable rate retardation. But one may ask, can there be rate accelerations? Because very many people think that if there is a bulge in RNA structure, so there is a strain, and this kind of bulged phosphodiester bond would be much more reactive than in a relaxed strand. And in principle, I think we may think about two things which might accelerate the cleavage, which might be, for example, behind somehow the ribosome catalysis. One is that if there is some kind of hydrogen bond network is formed around this region, which somehow facilitates the proton transfer from here to here, to the departing oxygen. This certainly will accelerate the process. Or if the initial state geometry would be great, so that this arrangement already is more or less collinear, and this is close enough to the phosphorus. These are the two things that I try to analyze. Okay, so we have tried to experimentally approach the problem in such a way that we have stabilized the RNA structure by methylating the 2' hydroxy functions everywhere except in one place, which still may cleave. There is still a hydroxy function which may attack. And we have inserted these scissile bonds to bulges, to hairpin loops, and so on. And what we observe is that if we put this reactive bond in a loop, in a bulge, the reactivity is the same as in a relaxed single strand. No acceleration, no retardation. The same here. Here a small acceleration. Only here when we go close to the double helical stem, on this side there is rate retardation, and also here when we are close to the double helical stem. Then some retardation takes place, but no acceleration. If the bulge is very small, there seems to be no strain which accelerates, but always the bond becomes more stable. So it is possible by molecular modeling to calculate different things from this kind of bulges, for example the geometry at the reactive site. And now you see that in all these nucleosides here, the geometry is such that the geometry is not optimal for cleavage, but the angle between attacking nucleophile phosphorus and departing nucleophile is 60 degrees. Not collinear, not 180 degrees, but 60 degrees. And the distance is quite long. Still, not really any rate retardation. Only what we see is that, so it means that the initial state geometry don't play too much role. but we can also calculate the stacking energy. And if there is an extensive stacking between bases, as here or as here, then the bond becomes stabilized. But it's the base stacking strength, not the initial geometry, which plays a role. We can see the same here. There is NMR data that the conformation of this bond is such that again the angle is 72 degrees, not 180 degrees. Distance is long. Still it is cleaved almost as rapidly as in a linear structure. If we change the place or the loop size, nothing much happens. A small acceleration when the loop becomes larger. Only in one case, a Mach acceleration, but I come back to this a bit later. The position within the loop don't influence much. Reactivities are more or less same. If we have a flanking end, here, when the leaving group is actually, this is now the leaving nucleoside. When it is base paired, the reaction is not decelerated, it's rather accelerated. But here, if the attacking nucleoside is base paired, then it is stabilized. And a bond opposite to a bulge, a small one or a large one, is also stabilized. It is similar to a bond in a double helical stem, which is stable. So in summary, there is a major change in conformation when the cleavage takes place, and it seems to be that the more flexible the structure close to the cleavage site is, the easier the reorientation is and the faster the reaction is. And the base stacking appears to be more important than the initial state geometry. But then I come to something which, to my opinion, is more interesting. So we have studied linear single strands, having no tendency to adopt any secondary structure. So we have used all computing programs. None predicts any secondary structure. We have selected a sequence so that they should be linear, relaxed single strands. And what we see, this is the basic sequence we have used. If we change this nucleoside here, this is the bond which is cleaved. If we have a UPA bond cleaved in 6 days, if we have GPA bond 160 days, CPA bond 270 days, APA more than 400 days. So the nearest their neighbors certainly influence. We don't understand exactly how they influence. But the UPA bond in this sequence seems to be exceptionally labile. But what is interesting is that if we make one base change very far away from the reaction center, here we replace this G with U, nothing else. The reaction becomes more than 10 times slower. Some other changes indicated in yellow result in small acceleration. And here is a quite amazing example. Still we have relatively same reaction site, CAAUAAC in both cases, but here UCU, here CCC. This is 30 days, this is more than 400 days. This is as stable as APA bond already. If we have a CPA bond similar thing. Change is very far from the reaction center, makes huge influence on the reactivity. So as you can see, RNA is a funny guy. So we tried to understand it. We did molecular dynamics simulations for the initial state first, to understand why it is so. And this shows that our calculations somehow work. So there is a conformation. So when it is calculated here about 2 nanoseconds altogether, so what happens is that it first seeks the correct conformation, the stacking energy, but then it stabilizes and starts to give some kind of average state. And we have calculated the stacking energy to different bases along these oligonucleotides indicated here. They are now exactly same sequences, except that one base is different at the cis bond. And as you can see, also the stacking energy remains very similar in all other sites except at the cleavage site. And it correlates very nicely with the reactivity. The one which is less stacked at the cleavage site is cleaved fast. And the one which is really stacked at the cleavage site is cleaved very slowly. Okay. Here an example where the change didn't influence on the reactivity because it was now far away from the reaction center. Also the calculation gives the same result. No change at the reaction site, change only here. But here a change far away in the chain which influenced on the reactivity. And now you can see that this change here is reflected to the base stacking at the reaction site. So when you make base stacking weaker here, it becomes stronger here, which indicates that base stacking in an oligomer is a cooperative process, not between just nearest neighbors. Does base stacking then really correlate? To some extent, yes. The correlation is not fine, but here is the initial state base stacking at the cleavage site. Here on y-axis and on x-axis the reactivity. There is some kind of correlation, but of course not very fine. Here is poly U. Now we see that some of the cleavages actually are faster than within a fully relaxed chain. This is easy to understand, that base stacking retards the reaction, but how the base stacking can accelerate the reaction. So we approach this by making a model for the transition state, or for the very reactive intermediate, the phosphorate intermediate. And we repeated the same calculations. First, this was optimized by quantum chemical calculations, and then we used these molecular dynamics calculations to get an overall view of the chain. And without going into the details, but it really seems to be so that in some cases, I think there should be one more. Yes, right. Now the cleavage site is here. These bases are stacked, and of course there is an overall stack, the whole thing. But now when the cleavage site opens, so when we come to this, when this opening takes place, it seems that the stacking here and stacking here can be enforced. So that actually the overall stacking energy is increased even when we have to destroy the stacking energy across the scissile bond. This is not very convincing. So here is the same correlation. And this is the extra stabilization which can be achieved in the transition state. And as you can see, according to these calculations, usually the numbers are higher here when we have exceptionally reactive species than here. But I admit that it is not very convincing.
Excuse me, what kind of errors are we talking about?
Sorry?
What kind of errors in measurements are we talking about?
It's the experimental errors. So the measurement errors are very small. So they are exactly there. What is the error in the calculation? So that is a philosophical question, because it depends on our model and everything. I can't tell it. But I think another thing which is quite important is the hydrogen bonding. And we tried to find some explanations by calculating the distances at the reactive site. But it seems to us that no direct hydrogen bonding actually is possible. So there are no good candidates. We cannot explain things. So the distance from the non-bonding oxygens or from the to the lower base moieties, for example, which might play a role, so seem to be too large to play a role. I skip this because. So, what I think, so long story, very shortly. It clearly seems to be so that stacking of bases plays a role. It can result in both retardations, accelerations. So this seems to be the apparent reason for the differences in the reactivities. But how?
Can I ask you what sort of interaction you are here what you here mean with the adjacent bases?
We have usually what we have studied is the strength of stacking so between the two bases flanking the scissile side. So this is the origin, but it actually depends on the overall base stacking pattern, because changes in base stacking far away in the chain will reflect to base stacking at this cleavage
site.
site.
But hydrogen bonds between the adjacent bases. Is it hydrogen bonds or?
No, there is no hydrogen bonding here. It's single strand. It's just stacking, vertical stacking of all bases, which has a contribution of van der Waals interaction and so on. So, but I think what really happens is that although we couldn't get strong evidence for hydrogen bonding or hydrogen bonding playing a role, I think that what the influence of this base stacking actually is such that the geometry at the cleavage site becomes favorable or unfavorable for appropriate hydrogen bond networking, and the real differences come from the fact how efficiently actually the leaving group becomes protonated during this process. So I think this was more or less what I wanted to talk, and I apologize. I know that this is not maybe very interesting for you, because this is mechanistically a very, very small detail which maybe helps to understand the action of ribozymes and so on, but I am not studying the RNA world, actually. Thank you.
So thank you for the talk and questions. Yes.
But you did a calculation where you showed that what you do shifted. So you sort of explained why the cleavage was more frequent in case you change the base for the cleavage, right? So presumably, when you did that in the calculation, there are different contributions to that. And you could see the different terms in which one that actually changed flow. If it was from hydrogen bond to from balls, or what type of interaction that contributed to that change.
So as I tried to say, so what we can see is that there is a correlation between the base stacking and the reactivity. But the base stacking in such a way that the base stacking is a cooperative process. So that is what we can say, that the base stacking influences. We couldn't, by calculations, get any real evidence for the importance of hydrogen bonding networking around the cleavage site. Was that what you asked? Or what did you...
Yes, but then what, yeah.
So I didn't quite understand your question, probably.
Yeah, well, I mean, it's just that there's a big difference in energy when you sort of lower the energy on one side and then increase it on the other side. And that was sort of an indication that the calculation sort of predicted this behavior, right? That they sort of experiments.
No, I think what we try to find is the correlation between the experimental observation and the theoretical calculation, which is totally independent of experiments. So the stacking energy was calculated purely theoretical, and the cleavage rate was experimental. And what we see is that when, if I make it a bit simpler, so when the stacking energy around the cleavage site is weak, then the cleavage is fast. And if the stacking energy around the cleavage site is strong, the cleavage is slow. But we cannot pay attention only on stacking in the initial state, but also in the transition state. And what we actually should correlate is the reactivity to the difference of these two stacking energies, one in the transition state and one in the initial state. And this correlation was not verified.
How are these different forms of RNA related to the biologically active RNAs?
So these has nothing to do with, no. This was just models for RNA.
But what about these different forms? Do they exist in vivo? When you showed several different forms and several alterations that we have focused on, do these alterations take place in vivo?
So what kind of, what do you?
Well, like in your figure, you showed as functional pH.
Yes.
That there take place alterations in RNA structure.
Yeah, of course that happens in the cell as well, but in the cell the pH is usually always more or less 7.
Yeah, and in the cell, well, it depends on the area of the cell.
Oh yeah, in endosomes it can be 5.
And it depends, of course, there are thousands of factors that regulate.
Oh yeah, but anyway, so we are, let's say, usually in physiological conditions from pH 6 to 7.5 or something like that, except in endosomes maybe 5. So what we did here, so our measurements was at a bit higher pH, more or less at pH 8. And the reason for that was that we tried to simplify the system a bit, because at pH 8 we have a single reaction. We have cleavage reaction only and no isomerization. If we go to pH 6 or pH 7, we would have the isomerization reaction taking place at the same time, which would disturb quite severely the kinetic measurement.
If we create such kind of such kind of RNA which a priori lacks the ability to form the secondary structure, and if we kind of start from this kind of short single-strand molecule and we gradually increase this molecule, then of course apparently statistically the stability of the closed molecule will be decreasing probably. Well, because the bigger thing is maybe probably the more vulnerable. But what about the stability of each concrete, let's say average phosphodiester bond between each concrete nucleotide in this increasing molecule? Will it be affected by increasing the length of the whole molecule?
No, not. I think what influences is the formation of the secondary structure, possibly or certainly the tertiary structure, but the length of the oligonucleotide as such at pH 7 doesn't play a large role. So let's say in real case in RNA there is actually very little pure single-stranded regions. It's only about 5–10%, and most of the regions are either stem loops or double helical stems or internal loops or this kind of peculiar structures. And what I would like to say is that most probably all these structures are somehow stabilized, more or less stabilized. But most probably also the folding to a tertiary structure, it will create hotspots into RNA, which is very well known that in an RNA molecule usually, even in transfer RNA which is small, you usually have 1 or 2 phosphodiester bonds which are cleaved very rapidly.
What is the rate? What is the order of rate of such cleavage? Let's say
here I showed that if we have just a linear strand, our temperature was 35 degrees, which is more or less the same as within the cell. So the cleavage half-life for the cleavage of a single bond, I think the maximum rate was something like 2 days, the half-life. Now you can say that RNA is not so stable. It is much more. It is much less stable. But you have to remember that I am talking about a single bond. If half-life for a single bond is 2 days, if you have 10 bonds, the half-life is 0.2 days. If you have 100, it's very rapid already, if there is no stabilizing effect, such as packing, which may result from double-strand formation, or proteins, which somehow protect the RNA structure, and so on. But a single RNA phosphodiester bond is not enormously labile, so it is reasonably stable chemical bond.
Coming back to the RNA world, can one imagine that some minerals or clays provide some stabilizing environment, structural stabilizing environment for the RNA?
Yeah, I think it's, to my opinion, it theoretically is possible, yes. On the other hand, I think if there are metal ions present, it certainly will make RNA usually much more labile, because metal ions quite efficiently cleave the phosphodiester bonds of RNA. So metal ions play a dual role. They are harmful for the stability of RNA in general, but they are quite important to obtain the tertiary structure which is needed when RNA functions as a catalyst. Yeah.
Yeah, just to connect to the question of RNA, well, I mean, I guess there are two problems you have to solve, right, with proteomics. You need to store the information and say it decays pretty quickly.
Yeah.
But the other problem is to copy it.
Yes.
And could you give me a sort of an estimate, because it seems still 2 days seems rather long if you compare to the problem of copying. Because say that the copying each base every 10 minutes or whatever, so, and if you have an accuracy of maybe 95% of your, you must have no
but you need a mechanism for this copy. So if you think that you have an RNA molecule which stores information, and you don't have proteins to help copying the information, it means that the RNA molecule itself must catalyze this replication or making a copy. And I think you mentioned that without any enzymes, so the fidelity of the copying will be very low, which I quite agree. So it means that if we want to make a copy of RNA without any protein, it must be a ribosome structure which catalyzes it. So you have to use part of your RNA to this chain folding. And then I think, yes, it is possible that it copies. But I think there will become a limit how long RNA molecule you can have so that it still is alive long enough, because most probably this copying by ribosome catalytic mechanism is not as efficient as the present ones, having the proteins to help. That it probably takes more time.
Yes, but my point is that I think that the copying fidelity will be a problem much earlier than the actual decay of the molecule itself.
Yes,
because you have to have quite accurate copying before you copy this long. I mean, this was pointed out by Ivan.
I quite agree, so that is not my field, but I quite agree that it may become a problem first, because you probably need quite efficient ribosome catalysis to copy this information. Yes.
You showed that if by misplacing a few or one nucleotide quite far away from the cleavage site, it had a great effect by more than a factor of 10. Is that now fully understood in terms of molecular dynamic calculations, or
it is not fully understood? I think it can be explained to a certain extent on the basis of differences in base stacking through the whole, but it is not fully understood. So this observation is quite new. It's about less than 2 years ago when we made these measurements, and these calculations are even more fresh. So I think we have to continue quite a lot in this field to fully understand.
But the molecular dynamics calculations do at least qualitatively.
Qualitatively they give an explanation, yes. Qualitatively, yes.
In this problem, I think you could draw at least some insight from formal language theory, context-sensitive grammars and languages.
Yes.
Because this is a linear
language. I'm quite sure of that. Quite a lot could be done by computing.
We have the founder of this science in Turku University, academician Arto Salmo.
Yeah, but
Who has also written the book of DNA computing.
Yeah,
I suggested.
I am just hopelessly weak computer.
Well, it's an interdisciplinary.
So I am dependent on collaboration field, frankly speaking.
So I think that just like for this talk, we still again end up in the situation that RNA stability has been one major problem in the early world if it was based on RNA function. So there would be a need some RNA stabilizing
So I am a bit surprised that as far as I have seen, no one of these big names talking about the RNA world, they don't touch the problem of RNA phosphodiester bond isomerization, which to my opinion is a bigger problem than cleavage.
Now would you explain?
Because if you have a 2' 5' bond instead of a 3' 5' bond, I'm pretty sure that the information cannot transfer anymore by the same ribozyme and by the similar mechanism as if, because the whole double helical structure is entirely different when you have a 2' 5' bond.
Has it been experimentally analyzed of if it would function to as any copying template, even for replication?
I don't know. I am not an expert of that field. I don't know.
Maybe we have to stop with RNA chemistry at this moment here and move to the RNA questions, catalyzed chemical reactions, bioorganisms, and we have continued